Can a simple touch cancel out the longevity benefits of fasting? Shocking new research from Michigan Medicine says yes. Scientists discovered that sensory inputs like touch suppress a key longevity gene called FMO2 in tiny worms. This longevity switch reveals the intricate connection between what you sense, your genes, and how long you live. The takeaway? Environmental cues profoundly influence lifespan. Specific genes act as longevity switches, and tryptophan metabolism is intricately linked to aging. Could this unlock drugs that extend life without extreme dieting? Stay tuned to find out. So exactly what did these researchers discover? Let me break it down for you in simple terms. Scientists at the University of Michigan used a tiny worm called C. elegans. Think of it as a microscopic guinea pig that scientists love to study because it ages just like we do. Here's where it gets fascinating. They found a gene called FMO2. Let me spell that out. FMO2. And this gene acts like a master switch for how long you live. Now here's the kicker. FMO2 is both necessary and sufficient for longevity extension when you restrict calories. What does that mean? If you remove this gene, fasting won't help you live longer. But if you turn this gene on, you can extend lifespan even without cutting calories. Think of FMO2 as the ultimate aging control button. It's the difference between manually restricting your diet for years versus potentially taking a pill that flips the same longevity switch. This isn't just worm science. FMO enzymes exist in humans too, and they're already turned on by several FDA-approved drugs. That means we might unlock longevity treatments without the suffering of extreme dieting. Now, you might be wondering, is this just speculation? Absolutely not. This research is backed by serious scientific credibility. First, this groundbreaking work comes from the University of Michigan Medical School, one of the top research institutions in the United States. The study was published on November 14, 2025. But here's what really matters. This research has been peer-reviewed and published in two of the most prestigious scientific journals in the world, PNAS and Science Advances. Think of these as the gold standard in scientific publishing. Before any study gets published in these journals, it goes through rigorous peer review, where top experts examine every detail. What does peer-reviewed mean? It means independent scientists, people who had nothing to do with this research, carefully analyzed the methods, checked the data, and confirmed the findings before giving it the green light. That's like having your homework checked by the smartest students in class before turning it in. This isn't just a blog post or a random claim on social media. This is research that has passed the highest standards of scientific scrutiny. When you see studies published in PNAS and Science Advances, you know you're looking at rigorous, reliable science. Now, let's talk about how this actually works, the biological mechanism behind this longevity switch. Think of your body as having a sensory switchboard that's constantly monitoring your environment. When you touch something or smell food, those signals don't just stay in your brain. They travel throughout your entire body. Here's the pathway. First, environmental cues like touch and smell activate specific neurons in your brain. These neurons then release chemical messengers called dopamine and tyramine. You've probably heard of dopamine as the feel-good neurotransmitter. But here's where it gets really interesting. These neurotransmitters don't stop at your brain. They send signals all the way down to your intestines through what scientists call neuropeptides. Think of these as molecular text messages traveling between organs. And finally, these signals trigger a cascade that influences how long you live. This entire communication highway is called the gut-brain axis connection. So what you touch and smell literally sends messages from your brain to your gut, telling your cells whether to activate longevity mode or turn it off. It's like your senses are constantly voting on whether you should age faster or slower. This mechanosensation pathway, the way your body translates touch and smell into biological responses, is one of the most intricate systems scientists have ever discovered. Let's zoom in on where all this longevity magic happens: your intestines. The FMO2 gene encodes a crucial intestinal enzyme called FMO2, and it acts as what scientists call a gatekeeper for longevity. Think of it as the bouncer at an exclusive longevity club. It controls who gets in and who doesn't. Here's why FMO2 is so special. It orchestrates complex metabolic pathways, meaning it coordinates how your body processes nutrients and responds to caloric restriction. It's like the conductor of a metabolic orchestra, making sure every instrument, every cell plays in harmony. Now, scientists did something fascinating. They ran two experiments: FMO2 knockout and FMO2 overexpression. Let's break down what they found. When they knocked out FMO2, basically deleted it from the worms, something interesting happened. The worms diverted tryptophan toward the serotonin pathway. Serotonin is that neurotransmitter you've heard about that affects mood. Elevated serotonin reduced exploratory behavior in the worms. They basically became less curious about their environment. And here's the catch: blocking serotonin restored their normal exploration. On the flip side, when they overexpressed FMO2, turned the gene up to maximum, tryptophan shifted toward kynurenine metabolites instead. This activated something called quinolinic acid, which also decreased exploration. And again, blocking quinolinic acid brought behavior back to normal. The key finding? Optimal FMO2 expression is vital. Both deficiency and excess can negatively impact behavior. It's a Goldilocks situation. Not too little, not too much, but just right. This research is essential for dietary restriction benefits and opens new life extension avenues. The manipulation of this pathway offers real hope for extending human lifespan without the suffering of extreme dieting. Here's where this research gets really surprising, and maybe a little frustrating if you've been trying intermittent fasting. Scientists have known for decades that dietary restriction and intermittent fasting can extend your lifespan. It's one of the most established longevity interventions in the scientific literature. Yet this new research on C. elegans worms reveals something unexpected. Environmental sensory cues can surprisingly negate these benefits. Now, what does that mean? Fasting benefits are often lost due to environmental triggers. Specifically, sensory cues like touch and smell of food can override the life-extending effects of caloric restriction. Think about that for a second. You could be fasting perfectly, but if you're constantly touching food or smelling it cooking in the kitchen, those sensory inputs might be canceling out the longevity benefits you're working so hard to achieve. Here's how it works. These environmental cues suppress the key longevity gene FMO2. Remember, FMO2 is that master switch for longevity. When sensory signals tell your body food is available through touch or smell, your brain essentially says, "We don't need to activate survival mode right now." The research shows that your physiological fasting state is directly linked to environmental perception. It's not just about whether food is in your stomach, it's about whether your senses detect food in your environment. This discovery profoundly challenges our understanding of caloric restriction. It emphasizes the deep connection between an organism's internal state and its environmental perception. The implication? True longevity benefits might require not just eating less, but also minimizing sensory exposure to food cues. Your brain and your metabolism are constantly in conversation with your surroundings, and that conversation directly influences how long you live. Let's dive into what happens when scientists remove FMO2 entirely, the knockout experiment. When the FMO2 gene is knocked out, the normal metabolic pathway for tryptophan is significantly disrupted. Remember, FMO2 plays a critical, unique role in C. elegans tryptophan metabolism. There's no backup enzyme that does exactly what FMO2 does. So here's what happens. The tryptophan flow gets disrupted, and it's diverted to serotonin synthesis. Think of tryptophan as a river that normally flows multiple directions. When you remove FMO2, it's like blocking one major channel. The water has to go somewhere else, and in this case, it floods towards serotonin production. The altered tryptophan flow is specifically diverted toward increased serotonin production, evidenced by a 40% increase in TPH1 expression. TPH1 is a rate-limiting enzyme, which means it controls how fast serotonin gets made. A 40% boost is massive in biological terms. This is where behavioral changes kick in. Elevated serotonin levels directly contribute to a significant reduction in exploratory behavior. The worms become more quiescent. Think of it as becoming more content, less curious. They're in a calmer, less exploratory state. Why does this matter? It tells us that FMO2 knockout profoundly impacts tryptophan metabolism, increasing serotonin signaling and thus reducing worm exploratory behavior. This isn't just worm science, it's revealing fundamental principles about how our metabolism controls our behavior. When one metabolic pathway gets blocked, others compensate, and those compensations have real consequences for how organisms interact with their environment. It's a perfect example of the interconnected nature of biology. Change one gene, and you create a cascade of effects throughout the entire organism. Now let's explore the opposite experiment. What happens when scientists turn FMO2 up to maximum? When scientists overexpress FMO2, the metabolism story flips entirely. Overabundance of FMO2 shifts tryptophan metabolism toward quinolinic acid, activating NMDA receptors and causing excitotoxicity. Here's the pathway. FMO2 overexpression alters tryptophan metabolism, increasing kynurenine-derived metabolites. Think of kynurenine as a different branch of the tryptophan tree. Instead of flowing towards serotonin like in the knockout, excess FMO2 pushes everything toward the kynurenine pathway. This produces elevated quinolinic acid, which is a neurotoxic, kynurenine-derived neuromodulator. Quinolinic acid is actually quite dangerous. It acts as an agonist of NMDA receptors. NMDA receptors, that's NMDA, are critical brain receptors involved in learning and memory, but when overactivated, they cause neuronal excitotoxicity. Excitotoxicity means neurons get overstimulated to the point of damage. It's like turning the volume up so loud that the speakers blow out. Quinolinic acid cranks up NMDA receptor activity, causing these neurons to fire excessively, which ultimately suppresses worm exploration. The researchers confirmed this by blocking NMDA receptors genetically. When they knocked out the NMDA receptor in worms with FMO2 overexpression, exploratory behavior returned to normal. This proves that quinolinic acid's effect on NMDA receptors is directly responsible for the behavioral changes. What's fascinating here is that the same gene, FMO2, can create completely different behavioral outcomes depending on whether it's turned off or turned up. Remove it, and you get elevated serotonin. Overexpress it, and you get quinolinic acid and excitotoxicity. This discovery highlights a critical link between neurochemistry and behavior. It shows how metabolic pathways in your gut can directly influence brain function and ultimately control something as complex as curiosity and exploration. Here's where this research gets really exciting. Scientists found rescue levers, ways to fix the behavioral problems caused by disrupted FMO2. Remember how we talked about FMO2 knockout leading to elevated serotonin and decreased exploration? Well, the researchers discovered they could reverse this by blocking serotonin synthesis. The intervention is simple. Block two enzymes called TPH1 and PAH1. TPH1 produces serotonin in neurons, while PAH1 produces serotonin in the intestine. When scientists knocked out these enzymes in worms with FMO2 knockout, exploratory behavior returned completely to normal. It's like flipping a master reset switch. On the flip side, for FMO2 overexpression, the rescue strategy targets the quinolinic acid pathway. Scientists found three intervention points: KYNU1, HAAO1, and blocking NMDA receptors directly. Here's the beautiful part. When they knocked out KYNU1 or HAAO1, enzymes that produce quinolinic acid, the worm's exploratory behavior was completely restored. And when they knocked out the NMDA receptors themselves, which are the targets of quinolinic acid, they saw the same rescue effect. This proves that quinolinic acid's effect on NMDA receptors is directly responsible for the behavioral changes. Targeting these pathways reverses behavioral changes. Normal behavior restored in knockout worms, behavioral changes reversed in overexpression worms. What makes this research so promising is that targeting tryptophan metabolism and kynurenine pathways offers a strategic approach to fine-tune both behavioral and longevity outcomes. Think about what this means. We might be able to activate longevity pathways without the negative behavioral side effects. By understanding these rescue levers, researchers are one step closer to developing interventions that extend lifespan while preserving normal curiosity, exploration, and quality of life. This is precision metabolic control, adjusting specific pathways to get the benefits of longevity without sacrificing the behaviors that make life worth living. Now, for one of the most important findings from this entire study, something that challenges how we think about longevity interventions. A partial decoupling exists between behavior and lifespan. FMO2 impacts both, but interventions restoring normal behavior don't always fully restore or sustain the associated lifespan extension benefits. Let me explain what that means. The FMO2 gene significantly impacts both C. elegans behavior and its overall longevity. We have seen how it controls exploration, curiosity, and responses to the environment. And we know it extends lifespan when activated by dietary restriction. However, and this is critical, interventions aimed at normalizing behavior often fail to fully restore or sustain the associated lifespan extension benefits. For example, rescuing FMO2 overexpression exploration mitigates some lifespan benefits, yet it does not fully suppress the extended lifespan. When scientists blocked quinolinic acid to restore normal exploratory behavior in FMO2 overexpressing worms, the worms did explore normally again. But they didn't live as long as the overexpressors with abnormal behavior. This reveals complex interplay and pleiotropy. The relationship is linked by tryptophan metabolism, showing that mechanisms for behavior and longevity aren't always perfectly aligned. This indicates pleiotropic effects, where one thing influences multiple, partially independent traits. Think about what this means for human longevity research. If we develop a drug that activates FMO2-like pathways to extend life, we might extend life but also change behavior in ways that reduce quality of life, decreased curiosity, blunted responses to danger, altered mood. The ideal intervention would be a Goldilocks solution. Extend health span and lifespan while preserving normal adaptive behaviors. The closest we've seen is the HAAO1 knockout combined with FMO2 overexpression. These worms maintain some longevity benefits, but display normal exploratory behavior. This research reveals a profound truth. Living longer and living well aren't always the same thing. The pathways that control how long we live are deeply entangled with the pathways that control how we experience life. Now for the million-dollar question. Does any of this matter for humans? The answer is a resounding yes. Insights from C. elegans FMO2 research directly inform human health, as key metabolic pathways are conserved across species. Here's what that means. The FMO gene family is structurally conserved from worms, FMO2, to humans, where we have mammalian FMO5. When researchers tested whether mammalian FMO5 could do the same job as worm FMO2, they found it can metabolize tryptophan in exactly the same way. This suggests similar longevity mechanisms across hundreds of millions of years of evolution. This research opens the door to something called caloric restriction mimetics, or CRMs. CRMs offer the appealing prospect of achieving anti-aging and lifespan extension benefits without the need for extreme dietary restriction. Imagine activating your body's longevity pathways without actually starving yourself. That's the promise of CRMs. But, and this is critical, there are potential risks and side effects. Manipulating these conserved pathways may lead to behavioral changes, such as altered sensory perception and decision-making observed in C. elegans. Remember how overexpressing FMO2 made worms indifferent to danger and less curious about their environment? Those aren't just worm quirks, they're behavioral trade-offs that could translate to humans. Think about what this means. If we develop a drug that mimics caloric restriction by activating FMO5 in humans, we might extend lifespan, but also dampen curiosity, alter how people perceive food smells, or change how they respond to stress and reward signals. Understanding FMO conservation and its pleiotropic effects is crucial for developing safe and effective human longevity interventions. The goal isn't just to help people live longer, it's to help them live longer while maintaining the mental sharpness, curiosity, and emotional balance that make life worth living. This research reminds us that the biology of aging is universal. The pathways that control longevity in a microscopic worm are fundamentally the same pathways operating in humans. And that means discoveries in C. elegans today could become therapies for humans tomorrow. So what does all of this mean for you right now? Let's talk practical takeaways, how you can design your life for longevity starting today. Environmental cues significantly impact our biology and health span. Our environmental cues profoundly impact our biology, even during dietary restriction. A mindful approach to fasting considers environmental context, managing sensory cues like food smell or tactile experiences to extend lifespan and health span beyond just food intake. First, environmental cues matter. Sensory inputs like touch and smell can negate the lifespan-boosting effects of dietary restriction by suppressing key longevity genes like FMO2 in worms. If this translates to humans, and the evolutionary conservation suggests it might, then your fasting benefits could be compromised simply by being around food. Think about someone practicing intermittent fasting but working in a bakery all day, constantly smelling fresh bread. Or someone meal prepping for the family while fasting. Those sensory inputs might be telling their brain that food is available, potentially switching off the longevity pathways that fasting is supposed to activate. Second, mindful fasting design. Beyond food intake, consider designing your environment to minimize sensory cues that might inadvertently cancel beneficial physiological states during fasting. This could mean avoiding the kitchen during fasting windows, not cooking for others while fasting, or minimizing exposure to food smells and textures. Third, sensory management strategies. Actively manage your surroundings, such as controlling the smell of food or tactile experiences to enhance the benefits of dietary practices for health span. Close the kitchen, use delivery services during fasting periods, or schedule meal prep after eating windows. The key insight from this research is that longevity isn't just about what you eat or when you eat. It's about the entire sensory environment surrounding your dietary choices. Your brain is constantly reading environmental signals, and those signals directly influence whether your longevity genes turn on or off. This is designed for longevity, intentionally structuring your environment to support the biological pathways that extend both lifespan and health span. It's not just calorie restriction, it's sensory restriction too. Before you go, we need your help making this research even better. Your insights fuel our progress in understanding longevity. This groundbreaking work from Michigan Medicine is just the beginning, and the next discoveries will be shaped by questions and experiences from people like you. So here's the question. Do you practice intermittent fasting or dietary restriction? And if so, have you noticed any unexpected challenges? Maybe you found yourself constantly thinking about food during fasting windows, or perhaps you work in an environment filled with food smells that make fasting harder. And here's what really intrigues me. What surprised you most from today's discussion about the FMO2 longevity switch and tryptophan metabolism? Was it learning that touch and smell can actually cancel out fasting benefits? Or maybe discovering how serotonin and quinolinic acid control our curiosity and exploration? Share your thoughts in the comments below. Tell us about your fasting experience, your biggest takeaway, or any questions this research raised for you. Your engagement fuels future discussions and helps us explore the most important questions in longevity science. Because at the end of the day, this research is about all of us, finding practical, evidence-based ways to extend not just how long we live, but how well we live. Let's figure this out together. For an in-depth understanding of this groundbreaking research, we encourage you to explore the primary studies from Michigan Medicine at the University of Michigan, published in Proceedings of the National Academy of Science and Science Advances. The first study, Rewarding Touch Limits Lifespan Through Neural to Intestinal Signaling, was published in the Proceedings of the National Academy of Sciences. This landmark paper establishes how sensory touch activates neural circuits that suppress FMO2 and reduce longevity benefits from dietary restriction. The second study, Metabolic Regulation of Behavior by the Intestinal Enzyme FMO2, appeared in Science Advances. This research reveals how FMO2 expression directly modifies tryptophan metabolism to control serotonin and quinolinic acid levels, which in turn shape exploratory behavior and decision-making in C. elegans. Both papers provide detailed methodologies, comprehensive data sets, and extensive supplementary materials. We've included the DOI numbers on screen so you can easily access these peer-reviewed publications and explore the full research yourself. These are truly fascinating reads for anyone interested in the cutting-edge science of longevity and metabolism.
Subscribe to BioTuber Online and unlock unlimited potential to improve every aspect of your health. Find real solutions for weight loss, blood sugar management, heart health, and mental resilience. Plus, science-driven answers for balancing hormones, boosting immunity, supporting healthy aging, optimizing digestion, managing pain, and preventing chronic disease. Learn to recharge your energy, enhance sleep, sharpen memory, and build strength through trusted fitness routines and nutrition plans grounded in the latest medical research. Explore topics like gut health, stress reduction, inflammation control, women's wellness, men's health, skincare, mindful living, reproductive health, bone and joint care, and integrative therapies. No matter your age or goal, this channel simplifies cutting-edge studies and expert insights, offering practical tips for total wellness, physical, emotional, and social. Join the BioTuber Online community for step-by-step guidance, actionable strategies, and daily motivation you can rely on. Subscribe now for evidence-based content and never miss an opportunity to thrive in every area of your life.